Welcome all. This afternoon we have Professor Klaus Müller for today's MOX colloquium. Professor Klaus Müller is Professor of Computer Science at Technische Universität in Berlin. He is the director of the Berlin Machine Learning Center, co-director of the Berlin Big Data Center, and of the Berlin Institute for the Foundation of Learning and Data, BIFOLD. He is a principal scientist at Google DeepMind. In fact, today, in this period, he's full-time at Google DeepMind. He's taking sabbatical from his academic services. He was awarded several prizes. I named just a few: the Olympus Prize for Pattern Recognition in 1991, the SEL Alcatel Communication Award 2006, Science Prize of Berlin 2014, Vodafone Innovations Award 2017, Hector Science Award 2024. He was elected member of the German National Academy of Sciences-Leopoldina in 2012, of the Berlin Brandenburg Academy of Sciences in 2017, of the German National Academy of Science and Engineering in 2021. He is external scientific member of the Max Planck Society from 2017. Also is in the scientific board of the Oberwolfgang Center. Is since 2019 highly cited researcher in the cross-disciplinary category. So it's a deep pleasure and an honor to have you here, Klaus. The floor is yours.
Thank you. Thank you, Laura, for your kind words. And it's really a pleasure to be here. I think it's my third time in Milan, so I feel I need to apologize for this because I think it's a wonderful place and with wonderful people, as you can see there. So I will talk a bit about different things that I'm interested in. There's a number of people who have contributed, so I put them here on the slides, but you also see them in the particular projects. First of all, some shameless self-advertisement, right? So BIFOLD is one of the 5 national AI centers in Germany. It's actually a wonderful institution because our government, in all its wisdom, has decided to make a permanent funding for a center, which is very unusual for Germany. And I think it's very nice because you can do really long-term things. And I actually like to do long-term things. I have the pleasure to co-direct this with Volker Markel, who is a big data specialist. For those of you who are using, for example, Apache Flink, this comes from Berlin, and that's from Volker. So having said this, that we have this center, we always have open positions. Just mentioning it. And I think Berlin is not so bad. Yes. Okay, good. Okay. So what am I talking about today? Well, I'm not sure whether this will. Okay, so I will talk a bit about explainable AI, which is something that I've been contributing to for the last 15 years. So this was like one of the first papers in this field that came from my lab. And it's now becoming very big. I will talk a bit about applications in pathology, medicine, histopathology, and I will talk a bit about quantum chemistry. And if time permits, there's some interesting video that I would like to show. Okay, so here's a paper. So you see many co-authors, and it's about histopathology in the context of breast cancer. So let me give you the background. So about 15 years ago, the husband of my sister died of cancer. He was a very nice guy. And I was very upset as we all are if we have our losses. And, you know, at that time, I was mainly working in neuroscience from the medical perspective. And I decided that I would like to have my own vendetta in terms of cancer, right? And I started a cancer program where I don't know, I'm not a medical doctor, but I can contribute in the sense of a machine learner, helping to make sense of data that exists. And there was a person, Frederick Klauschen, who's the last author on this paper. He's a very unusual professor. He's a professor for pathology, he's a medical doctor, and a physicist. So he can talk math. And that is, you know, we have several people like that in the medical school, and it's fantastic because you can really engage and talk about the details, and this way also gently get introduced to the medical field, right? Okay, so the first idea was to say, how can we use machine learning to, you know, diagnose or classify in the very simple sense. So here's a slice that comes from a biopsy, and you can, you know, if you're, you know, a specialist, you can make sense of this, right? And this is what the doctors do all the time. These slices come from surgeries often, and the surgery is stopping while the pathologist looks at this and wants to see, is it cut enough around the cancer? And then the surgery may proceed or may stop, right? And so then we said, well, okay, it's quite a slice. So you have a picture typically of 200,000 times 150,000 pixels. So it's about roughly 2,000 or 3,000 times a cell phone picture. Then you, since it's too much, you split it up into single smaller pictures, and then you would run it through a machine learning model, and the machine learning model would say cancer or not cancer, something like that, right? So we were quite happy because we got some, you know, nice performance, 90% or something like that, which is quite kind of nice. And I showed this to Frederick, and he said, boring. Okay, so then, you know, but we actually wanted to help, right? So what's non-boring, right? So what is non-boring is to say what exactly does the machine learning model do? Why is it saying this is cancer? What's the cause, if you want, or the association, or what is the explanation of what your classifier does? Okay, so that was the first time we asked this question in this context. And I feel I need to get rid of this here. Okay. So now, after the story, we will talk a bit about explainable AI, because that was the motivation. So people that use nonlinear methods, whether it's deep learning or any algorithm that you can think of, they would typically complain and say, oh, this is a black box. We cannot understand what it does. And this is not true anymore. And I will show you how, you know, you can go about this. And now I'm not talking about histopathology anymore, but I will be, you know, a bit more, you know, in terms of livestock, right? We talked about this over lunch, but anyway, so you assume that you have a model that is able to distinguish between roosters and buses and Dalmatians and God knows what, right? The typical computer vision exercise. And this model is very good and it can generalize. So then this is typically a deep learning model. And so you would give a new image to the model, and then the image, the model would say, well, you would classify this as a rooster. Okay, nice. So then the question is, this thing here is a totally nonlinear beast. Some very complex function class. So how can we, explain? Meaning, how can we go backwards through the decision-making process and explain what the model does in terms of the pixels? So that would be one way of explaining. And for this, we need to solve a mathematical problem that was so far unsolved that period of time before our paper in 2015, namely, putting this nonlinearity upside down. Okay, so which means that we go backwards through the model and then get something which we call a heat map, where every pixel gets a certain weight, how much it contributes to the classification. So you can see green is neutral and red is actually speaking for roosters. Okay, so you can just eyeball this and you see the red part is the rooster's comb. So the neural network that has learned about roosters and dogs and whatever thinks that this particular new, never seen before image is a rooster. And you can go backwards to the model and you can see, okay, it's the rooster's comb. So for me, this makes sense. Okay. So the question is how do we do this? Because obviously we have a nonlinear function. And so the first thing that we do is we go forward through the model. Okay, so we have some picture, and then we have, sort of the input layer and a bunch of hidden layers, and if we have many, then it's a deep model. And there's some nonlinear transformations and some shearing and warping and clipping, and in the end, there's the output layer, and the model says it's ladybug, right? Because here it's a ladybug. So how can we move backwards? And the idea is now the following. So what our algorithm, which is called layer-wise relevance propagation, is doing, it's taking what we get from the forward path, right? And then we call this relevances. So just to make this clear, we have frozen the model. The model is not learning anymore. All the weights are fixed. So now we have, from the forward path, we know it's a ladybug. So we have the relevances for this decision in the output layer. So now we initialize the process, and then we move backwards, because we have to iteratively go from layer to layer until we have the input features. So here is one neuron, in an earlier layer, and we compute the relevance of this neuron by taking the activity from the forward path of this neuron for this picture, and then a summation of all the relevances with the weights that the model has, appropriately normalized. Now, this looks a bit arbitrary, and a lot of people have, in the meantime, put up formulas like this. So the question is there anything, mathematically that you can say about this? So just before I say that, it's clear if we have the relevances in this layer, we can do the same game again for this node and go backwards. Okay. So, and there's one thing missing, which is that the relevance needs to be conserved. For every layer, the sum of the relevances need to stay the same. And now I actually would like to have a sheet of paper. Does anybody have a sheet of paper? Wonderful. Okay, so why is what we're doing something meaningful? Okay, so think about first what does a neural network do? In some space that is spanned by the pixels in RGB, so it's a high-dimensional space, we're having some decision function that is nonlinear. Let's assume that this is a piece of paper, right? Okay, it's nonlinear. I don't overdo this nonlinearity now. And so now, you know, the first thing you could say, well, how about describing this function by a Taylor expansion? We take one root point, and then we take a Taylor expansion, which in high dimensions, we have to compute an awful lot of moments, right? You know, whether this is unfeasible or not, let's not think about this for the moment. But it's a complex function, and we have to do the Taylor expansion. It's hard, and it's unfeasible practically because you have finitely many data, you have many dimensions. So this is not going to work. But what we could do is we came up with a deep Taylor decomposition. It's deep. Anyway, so so the idea there is to say, how about doing a linear Taylor expansion? Okay, but not around one root point, but around many root points, because local linear is global nonlinear. So it's just putting, you know, if you think about the decision surface, you put a bunch of linear functions, and then, you know, they're attached to some root points, right? And it turns out that if you do that, you can first of all prove that this is equivalent to a global Taylor expansion, a nonlinear one. And you can also show that it's easy to compute the root points, and it's also easy to compute the local linear approximations. And you can go one step beyond that. Every root point is, so to say, related to a particular neuron, right? If you think about what a neural network does, then, you know, a neural network gets some input, and then it puts some nonlinearities, often ReLUs or some nonlinear function, and it places them somehow, somewhere, right? And so you're basically just using that, what you have learned, and then, you know, placing, you know, the linear approximations there. And if you, you know, read a longer paper with some math, like by Grégoire Montavon and myself in pattern recognition, then you can see the proofs, and, you know, you can see that this is not an arbitrary formula, but it's a formula in the spirit of this Taylor expansion, okay? And so the nice thing about this theoretical framework is that you can not only apply it to some arbitrary neural networks, you know, deep learning models, but also to transformers, to LSTMs, to anything on the planet, so to say, okay? So then we are ready, you know, to show cat pictures, right? No talk should be without cat pictures. So we have now some models that are very nice and can distinguish cats and coffee and whatnot. And we have, you know, neural networks on one side and kernel methods on the other side. We are analyzing models that have been trained. And you can see, you know, cats are sometimes cats because of the fluffy part here, or the mustache, or, you know, of course, the coffee, as I could experience, is, you know, considered coffee and recognized coffee because of the crema, all right? So, you know, so makes sense. So these models have learned something meaningful, okay? It's always unseen data. Okay. So now, so there's layer-wise relevance propagation. We have this deep Taylor framework, and depending on whether we have a pooling layer or convolution layer or, again, LSTM, whatnot, according to the deep Taylor, you know, way of constructing things, we have to change the rules slightly. Okay, so but that is part of frameworks that you can use. But if you don't use the right LLRP rule, then you're not optimal in the sense of the framework, which means that your results are not so good. So, okay. Now, we've learned that we shouldn't complain about deep learning to be black boxes. And we have understood that there's at least one algorithm, the LRP, which I explained, which comes from our lab, is able to open the black box. There's, of course, many other explanation algorithms. It's a big field now. There's more than 1,000 people working on this. There's own conferences now on this. So, but let's now think of what we can do with this, what we can learn with it. And so what we typically learn in our class is that a standard machine learning model will take some data, then it will train a machine learning model such that it generalizes. You do model selection for that, and you do all the necessary things that you also learn in class. So the generalization error is the meaningful, important thing. And that's still true. But the question is it telling you everything, right? And I will just show you a nice little experiment that we had in CVPR. CVPR is the computer vision conference that, is the go-to place. So in CVPR, often people develop new architectures and new frameworks, and they show that they are good, right? So the typical thing is that you have some benchmark and some, here we would take an antique benchmark, which is Pascal VOC, which served the community for about a decade. And now it's not even ImageNet anymore, but let's take this aside. You have some models, and these models, you report what the numbers are out of sample in the way that people typically do. And typically, if your model is really great, and so it's better than the other models, then perhaps you get a CVPR paper. And I'm saying this in a half cynical way, right? So, of course, computer vision is doing much more than, writing tables, right? But there, you would have this concept, this type of result. And so now you can look in the table. And you can see that model Fisher and model DeepNet, they're basically having the same performance in terms of their generalization error. There's no statistical significant difference. So now we have explainable AI in order to check how the model solves the problem. And here is an application of the model to new data. And this is a nice picture of a horse. And both models say, yeah, it's a horse. Both models are right. So then you can ask, what is the model? Why is the model saying that this is a horse? So the deep learning model says, well, it's a horse because of the object, because of the horse and the rider. In particular, the horse's butt is very characteristic. And then you have the other model that basically is all over the place. Horse's butt is a bit, right? But this is a particular salient part, okay? So I will just explain what is there. So it's actually a copyright tag. It says Lothar Lenz Pferdefotos.de. So it's just a horse picture tag. So it happens, and of course, these big benchmark data sets are never looked at by people because there are millions of data points to look at. So you have in this horse class, you happen to have 50% of the pictures roughly that have this copyright tag. So in order to classify the horse, it is a completely valid strategy to say there's a copyright tag and that's the horse, right? So, but if we are talking about buses and cats and horses, maybe this is not so important. But if we're talking about medical images and diagnosis, it is important that we are, we understood the problem. The network has understood what it should learn, right? So when I showed this to my wife at some point, she said, well, what are you so worried about? And I said, yeah, the model is cheating. And she said, no, it can read, right? Anyway, I mean, so because this is so blatant, right? And it happens so often, we gave this a name. And we named it after this horse. So this horse's name is Hans. It's a Berlin horse around this, 1904 or something like that. And this horse allegedly could do math, right? So the horse's owner here on the picture, points at some part of the table, and there's some math exercise like 2 + 4. And then the horse would take its foot and tap as many times, in this case 6 times, on the board, and the horse was never wrong. So the journalists, of course, they were totally happy about this and said, horses can do math now, finally, right? And then some people with more maybe common sense came about and said, well, perhaps the horse is reading its master, and, horses are very sensitive, they can read humans. So even if they exchanged the human by a random human, the horse could still get this right. But the strategy was not doing, math, but the strategy was reading the human. So therefore, the horse was also called Clever Hans, right? And so therefore, this is the Clever Hans effect of a model. And I would say that, a large part of the models out there are of the type Clever Hans. And there's absolutely no excuse not to apply explainable AI. So if you do something meaningful, you have to do it, because otherwise, God knows whether there's a Hans lurking, right? Okay, so explainable AI can help to reveal bugs in the strategy, something that the generalization error doesn't tell you, right? It can discover data flaws, right? That's also an important thing, because that's a flaw in the data. It's a copyright tag. It's an association that is, somewhat random. And it also can help you to get insights in the scientific domain. Okay, so let's go back to this example, because, fast forward, we developed this explainable AI techniques, LRP. Fredrik is also a co-author of this paper. And by the way, just to say that, when we first submitted this paper, the reviewer said, no, explainable AI, nobody needs this. Okay? And of course, it's nonsense, right? And I think and nowadays people know that, but it's sometimes you submit stuff and you get, replies by the reviewers that you don't like. And sometimes they're right, okay? Let's keep it like that. But sometimes this is just nonsense, right? It's just something that puts everybody outside their comfort zone. And it doesn't matter that, it doesn't mean that your idea is bad, right? So you have to stick to it. Anyway, so now we're classifying again. And now we do the heat map. Okay, so I will show you a bit more details. So that's the cancer tissue, and that's the heat map, and that's the overlay. So you can see some of the cancer cells that are nicely marked. Okay. So what does it help? So it means that we're now having models where we can see whether the strategy of the model, not only the diagnosis performance quality, whether the strategy is the right one that makes physiologically sense, okay? And so now we can start playing with this tool, and we can not only classify cancer or non-cancer, but we can also classify lymphocytes, stroma, cell types, the whole program. And then we can get heat maps. So we put in the histopathological image, and we get these heat maps. Now we were becoming a bit adventurous and said, well, how about, in some of the data for some of the patients, you actually have protein expressions and copy number variations and a lot of biological data. So, but only one data point per slide, right? So you don't have it spatially resolved. So you have about 60,000 different protein expressions, something per slide, but not spatially resolved. So you can now start saying, well, how about doing regression to all these parameters? Okay, and most of the cases you can't predict anything because there's no biological meaning in this anyway. So it's a random, prediction that you can only make. But for example, if you have cancer tissue, and then typically the growth factors, impressive, and then you can ask, can we predict this? Yeah, you can do this. And then we can say, well, and how about the explanation? So then we can get a spatially resolved map of something which was not spatially resolved before, right? Now we did this, and you can see some of these maps. And then of course, the standard procedure, you send it to a journal, and then people say, wow, no, it's a bunch of machine learners, they have no clue. We don't believe this, right? And so, this kind of spatially resolved heat map of different properties, molecular properties. It's not true, they said, right? And they force us to do experiments. So because you can actually take the pathological slice, and then you can take the machine learning prediction for a certain staining. And here's ekaterine. And then we can say, well, okay, so here's the heat map that we get for the different slices. And this is the immunohistological staining of that slice. From the perspective of biology, this is quite remarkable. And so we could, basically show that indeed it's true, right? And of course, having said this, it's a step in a certain direction. So it's not the ultimate truth, right? But it's something where we can start data-driven hypothesis building. And of course, we cannot believe in these hypotheses unless we have done some interventions. But that's also possible. So it's actually a very interesting tool which we now have. Okay, so for insights. Okay, and here's a bit of this from the pathologic perspective, but also the XAI perspective. Now I must. So this was basic research, but I think it's an important role that machine learning and modeling can do is to not only say that some predictions are of medical value, but actually to put something into a clinical context. And so a lot of, maybe there's 15,000 machine learning papers that claim something in medicine, but stuff that really goes into clinical use is about 3 dozens, right? And so I was very happy about our one paper, and now we have more, where we actually are in clinical use. And I would like to share this with you. So, you see again some of the names that you've seen before. And you have 2 cases here. And I'm just telling you about the case. So you have a patient that presents itself with a squamous cell head and neck cancer. Cancer is removed. And then 5 years later, again, hospital is revisited. It's again a squamous cell cancer in the lung. And the diagnostic question is it a primary lung cancer or is it a metastasis? If it's a metastasis, then the survival prediction is very bad, 3 months maybe. And in the case of primary lung cancer, it can be removed and then the person can live for a long while. So the problem is if you look at the histological images, that it's a 50-50 call because both are squamous cell cancers. So under the microscope, they look not distinguishable, right? So all these cases are very difficult cases. They are also not rare, and they always go to the tumor board. So there's a group of specialists that try to take all the information that is available about the patient and then make a decision. Okay, so here's our approach. Okay, so we take DNA methylation. So you can see the DNA, and then there's some methylation, this CH3. And so if you look at your DNA, in fact, you can do this easily by just cutting a piece of tissue and then putting this into a machine, paying $300, and then you get an 800,000 dimensional vector. Okay, so but what does it tell you? So it's basically a book of your sins, you know, what you have done wrong, but also it's also a book of the history of a cell, right? So the methylation tells you where the cell comes from. That's a very interesting aspect of it. So now we can take this DNA methylation data, and we have, you know, not many cases, and then we can, you know, make a dimensionality reduction. We can train a model. It can be a decision tree, or it can be a neural net, or it can be something, right? And then we can have a validation cohort that we haven't looked at, and we can apply our model so that we have a, you know, some understanding of how good we are. And we have taken public data for this. So the classical random forest would be basically at a threshold accuracy of 99 point something, right? Then, you know, you would have the number of predictable cases 43%, which means that if you know, the model is not sure in many cases. And if you're not sure, then you rather reject the case. Okay, so which means that 57% of the cases are rejected. They cannot be judged by this method. So now this is our method. And it's quite surprising. At the same threshold accuracy, essentially, we have about 92% of the cases predictable. Think about the medical expert, you know, looking at the histopathology would just have a 50-50 call, right? So this is very, very useful for diagnosis. So when we actually submitted the paper, the word spread, because this is so unusual that you can do something that is very useful in practice. So in the medical school, you know, people talked about it. And, you know, already before the paper got its first reviews, there were several cases in the hospital, and my colleague Frederick was contacted by them and said, yeah, we have heard something, and perhaps you can, you know, apply your method to our samples. Of course, clearly, it's always going to the tumor board. So it's one piece of evidence that is on the table. And I think that's all it is, right? And I think by the time the paper had appeared, It was already 30 cases just within Charité. The interesting thing also is that, you know, this was on public data. And then at some point people said, well, you know, we have some in-house data. So which, you know, you have not seen. How about applying it there? And I said, I forgot how many cases that there were, maybe 60 or so. And so we had the tissue sample, so we could extract the methylation data and so on. So, and I think we had something like, if it was 50 cases, we had 49 cases right, according to the diagnosis that was historically taken. And then I said, well, you know, statistics tells you, you know, that's about it, right? And they said, wait, no, let's not be so quick. Let's reanalyze this case. And they looked at the medical files and they looked at the case and they found that this was a misdiagnosis, which made us very happy, but you know, eventually statistics wins, right? I mean, okay, so I have been talking too long about this medical case. Let me just quickly talk about quantum chemistry. So now for something completely different, right? So in another sabbatical that I had in 2011, I was invited to a place called IPAM. As mathematicians, you should know this place. It's the Institute for Pure and Applied Mathematics. It is at UCLA. It's a wonderful place because it runs, first of all, workshops, but also long-term programs. And these long-term programs are typically 3 months. So typically the setup is that, you know, some 15 or so faculty go there and they bring a postdoc or a student. And then this is like a merry crowd of, you know, maybe 40 or 50 people that are together and they are in the same building and randomly interact and get stuff done. And usually if you do a sabbatical somewhere, then everybody around you is busy because they're not in a sabbatical. But there, you know, everybody was on a sabbatical. So they were ready to just try something, right? And I learned that this was a program on chemistry and in particular on trying to understand the chemical compound space. And so my history is that I did my master in theoretical physics. So I was trained to do quantum mechanics, quantum field theory, strings. And then I moved to computer science and did neural networks. So now I came there and everybody was talking about the Schrödinger equation, which I, you know, remembered. You know, that's the Schrödinger equation and Schrödinger. And it's unfortunately, in all its simplicity, it's a beast. Because if you have more than one electron, then the interaction kills you, right? So it means that you cannot properly solve it. You only approximately solve it. And so basically what people have been doing is they have been creating methods like density functional theory in order to solve this problem. And that got Nobel Prizes because it's an approximation of that is useful for chemists. And so when I saw this, I thought, well, how about instead of, you know, simplifying and then doing approximation, how about treating this as a stochastic problem in the first place? So instead of basically, you know, that we are taking some data that we're generating with, say, DFT, density functional theory, and learning it. So we're learning to shortcut the Schrödinger equation. We're learning to predict the outcome of the Schrödinger equation. So which was a bit heretic, right? I must say. But the quantum property of matter are important, right? In everything, if you want to understand how drugs work, if you want to understand how chemistry interacts, if you want to understand materials, it's all about quantum chemistry. It's not. And the compute that you need is insane. So most of the quantum chemistry people and the climate people are really the most supercomputer users still. The typical thing is for a small molecule, you take about 4 hours of compute to get its quantum properties. For material, 4 months, right? So it's a bit of expensive data. Okay, so how do we represent this? Because we need to put it into a machine learning model. So the first way of representing things was to say, we take the molecule, so we have the nuclear charges and the location of the atom. And then we take basically the Coulomb force, write it in the matrix, right? With something on the diagonal. And then if we want to compare the similarity between objects, which you always have to do in machine learning, then, we do take a Frobenius norm, right? And then I insisted in doing the most simple thing that I knew, which was a kernel ridge regression model, where you put your Frobenius norm into a kernel in an exponential, and then you solve this nice, you have this kernel ridge regression predictor. So these are your parameters. This is the novel molecule that you see. This is your training data, and this is some threshold, and you regularize a bit, and the solution is basically a piece of cake, right? It's just a matrix inversion. So interestingly, this worked, right? So in the first moment, our first paper on this was said that we have 10 kcal per mole, just taking this Coulomb and kernel ridge regression. Then a bit later, we could also take a neural net, and a bit later, you could take transformers and everything. And in the end, you have about 2.2 kcal per mole, which is chemical accuracy, or in fact, below chemical accuracy. So this 1 kcal per mole is what makes chemists happy, because they can do something with it. Okay, so now we have sophisticated models. This is like our tensor neural network, or we have also transformers, or whatnot. And then we can ask, maybe there's a clever Hans, right? So what are we actually doing? What is the model doing? Because the model is trying to, this neural network, other than the kernel ridge regression model that just takes the Coulomb representation, which is a very simple physical representation, the neural network can learn from a data set how to represent an atom in its chemical environment, right? And then we can start visualizing this. And for example, with a trained model, we can ask, say for this benzene, if I were a hydrogen atom, what would be the chemical potential, and I put this in quote, that this would feel? And we could ask our model what the model says to this, or the carbon. Basically, where would you bind, right? And if you can do this for all the models that we have looked at, and it's giving some chemically meaningful things. Same for materials, right? And then we can ask a more philosophical question that comes back to the chemical compound space. So, humanity has been, navigating from the stars because they had a map. You could travel the oceans and navigate, our planet. But where's the map of chemical space? And chemical space is vast. So the number of small molecules is higher because of the combinatorics than the number of atoms in this universe. So it's huge. So in this vast space, how can we orient ourselves? because, you know, some molecules have certain properties, and how can we, you know, perhaps get similar molecules of similar properties or slightly different properties? How can we navigate? And so just to say this again, standard DFT would give us 3 to 4 hours, 5 hours per molecule, 4 hours for materials. So navigating is a bit difficult, right? You know, machine learning would give fractions of milliseconds. It's still difficult to navigate such a large space, but we can at least look at some subspaces. So one of the motivations is to do that, right? And it's a huge program, right? And I think I'm already going over time quite heavily, so maybe I will almost stop here. And I will just, perhaps, if somebody asks what curly is, you know, talk about this. But before I can, I just need to conclude. There's a need for opening the black box. Certainly, if you want to do AI for the sciences, clearly in medicine. Clever Hans is everywhere. Interestingly, and there's a new paper coming up, you know, everybody is talking about foundation models these days. And the training of foundation model is with self-supervision. So, and the question is there Clever Hans also in unsupervised learning? And unfortunately, the answer is yes. So interestingly, the reason for this is, while in supervised learning, it's mainly the quality of the data, in unsupervised learning, it's the quality of the algorithms. In other words, the statistical algorithms of representation learning, they are actually overfitting. And that, I think, it was a very interesting insight. And the problem in this is, if you have a Clever Hans in your foundation model, and you know, we've looked and seen many, then you inherit this at every downstream task. But it can be avoided, okay? You know, it's a recent paper. So XAI is available for all sorts of methods, unsupervised learning, transformers, graph neural networks, whatsoever. It's essential for understanding. And we need to contribute to medicine, and we need to do this in a meaningful way. It's not enough to just give tables. That's boring and useless. We need to understand the mechanisms. We need to contribute to that. And, you know, it starts with a boring table, but then we have to go and understand things. So the boring table per se is not boring, but if we stop there, then it's boring. I think one aspect that I also want to mention is that often people complain that about, you know, foundational research. And I think it's really a wrong complaint. They say we are not transferring enough to applications to industry. And in particular in the field of machine learning and modeling, this is nonsense. So when you are building a new car, right, and you have a nice invention, it takes about 10 years until your invention is in the car. But in machine learning, depends on where you work. You know, a new idea which is genuinely mathematical can be in practice and making money within 2 weeks. So that means that we have to really think about new things and new foundational things. This is my conclusion. Okay, thank you very much.